How will gene editing change dementia treatment? How will gene editing change dementia treatment? This is a question that many researchers and families affected by dementia are asking. Gene editing has the potential to transform how we approach dementia by targeting the genetic roots of the disease. Current treatments mainly focus on managing symptoms like memory loss and cognitive decline. However, gene editing aims to correct or modify the specific genes involved in dementia's development, including Alzheimer's disease and other forms like vascular dementia and frontotemporal dementia. Recent advances in gene therapy and gene editing technologies, particularly CRISPR-Cas9, offer exciting new strategies. For instance, gene therapies are being developed to reprogram brain cells to restore healthier functions. In animal studies, researchers have shown that gene therapy can reverse memory loss in Alzheimer's mouse models by changing gene expression patterns in the brain. This method focuses on preserving cognitive function by influencing how brain cells behave, rather than just targeting the harmful protein deposits associated with dementia. CRISPR-Cas9 technology allows scientists to edit DNA with precision. Researchers are studying how to directly target genes that produce toxic proteins like amyloid beta and tau, which build up abnormally in Alzheimer's disease. While no human clinical trials have been conducted yet, preclinical studies in animals suggest that gene editing could prevent or slow disease progression by correcting mutations that lead to these harmful features. Moreover, computational approaches have identified existing FDA-approved drugs that can reverse gene expression changes linked to Alzheimer's. This means that modifying gene activity could have therapeutic effects. Combining gene editing with these drug therapies may enhance treatment effectiveness in the future. However, significant challenges remain before gene editing becomes a standard treatment for dementia. Researchers must develop safe and efficient methods to deliver gene editing tools across the brain's protective barriers. They also need to ensure that therapeutic genes are stable and expressed long-term. Thorough evaluations of safety and efficacy through clinical trials are essential. In practical terms, gene editing could lead to personalized treatments for dementia patients by targeting their specific genetic profiles. This could potentially halt or even reverse memory loss and cognitive decline. While these advances are promising, they are still largely in the research and preclinical stages. Current dementia care continues to rely on managing symptoms, and gene editing should be seen as a future complement rather than a replacement for established medical care. In summary, gene editing holds the promise to fundamentally change dementia treatment by addressing the root genetic causes. This offers hope for therapies that could slow, stop, or even reverse the disease process. However, more research and clinical validation are needed before these approaches become widely available. Staying updated on dementia research, resources, and advocacy will be crucial as we navigate this exciting frontier in medical science. Always consult healthcare professionals for personalized medical advice and treatment options.